Welcome, everyone. It is my pleasure today to welcome our CCAM seminar speaker for today, Professor Lior Sepunaru of UC Santa Barbara's Chemistry and Biochemistry Department. And I should have asked you if I pronounced that correctly beforehand. I'm sorry.
That is correct. And Sepunaru, by the way, is from saponification, which means soap maker.
Oh, okay. I learned something new. So Professor Sepunaru joined UCSB as an assistant professor in 2017, where he leads the electrochemistry lab. He received his PhD in 2014 at the Weizmann Institute of Science in Israel, studying solid-state bioelectronics in the groups of David Kahn and Mordecai Sheaves. Later, he went to the University of Oxford, where he was a postdoctoral Marie Curie Research Fellow with Richard Compton. His current research interests include the interface between biomolecules and conductive materials, electrocatalysis at the molecular level, nanoparticle biosynthesis, and applications of liquid cell TEM to these and related areas. And because of this exciting synergy of research interests, we're delighted to have him today as our seminar speaker. And also he mentioned that he's very happy to welcome questions throughout the talk. Is that right, Lior?
Absolutely correct.
Okay, so all of you should have the ability to unmute yourselves, and I will also try to monitor the chat as we go along. So we're looking forward to your talk. Take it away.
Thank you, Professor Kopp, and thank you for the kind invitation. I think that we should have, or we can have fun together. I want to emphasize Stacy's note about any questions. Stacy mentioned that I am coming from Israel. In Israel, the way that we evaluate a good talk is that if a talk is not actually finished because there is a lot of questions coming during the talk. In the UK, after that, when I did my postdoc, it's the opposite, and the audience is silent. So what I would like to do is to encourage you to ask any question either via the chat or directly to me, which is preferable, during the talk. I have planned the talk to be around 40 minutes, 45 slides, so we have time for that. Today's topic will be about a new term that I believe we coined in the literature just recently, which is Electro Assembly of proteins. And this work would never been done without the beautiful collaboration that I have with Mike Gordon from ChemE and Dan Morse from biology. So the topics for today will be as follows. Because we will talk about electrochemistry, I'm well aware that many scientists and chemists and material scientists are not aware fully of what is electrochemistry. So I will briefly explain why should we care about electrochemistry. After that, I will talk briefly about other aspects in my lab that are connected to single entity electrochemistry, the ability to probe single objects one at a time. And then I will go to the main topic, which is protein electro assembly. And I will try to connect the single entity electrochemistry just at the end of the lecture. So about electrochemistry and who cares? The picture that you're seeing here is a picture of an old potentiostat. And there is a reason that I share with you this picture, because it's ugly. And that's how typically people think about electrochemistry. In our lab, A, we have state-of-the-art potentiostats that do not look like that. B, we can build our own potentiostats and preamplifiers. Why should we care? Well, one quick motivation will be to present these three gentlemen that are the Nobel Prize winners of 2019. What you're seeing here is Whitman, Goodenough, and Yoshino, who developed the lithium-ion battery. If that's not enough motivation for you, you can actually take this kind of technology and become extremely famous. And with a single tweet, you can change a bit the world economy. But I would like to be a bit more serious about the scope of electrochemistry in general. If I talked about, at the moment, quickly about the fact that you can use chemicals as a way to produce energy, you can do the other way around. You can use electricity in order to derive chemical reaction. And what I will try to do today is to show you one aspect of that and maybe a bit of deviation from this dogma by talking about electroassembly. What you're seeing here is the wide applications of electrochemistry. Energy is something that I mentioned before. Sensors is another notable application. The biggest example will be starting from the 1980s, the development of OH Hill of the glucose sensor that comes from the electrochemistry, and a lot of personalized medicine that are at the moment still attempts based on electrochemistry. On top of that, there is a huge buzz starting 6 years ago about electrosynthesis, electroorganic synthesis, that is becoming an extremely hot topic. That is that you can synthesize molecules with mild conditions with electrochemistry. Separation, of course, synthesis of aluminum, which is a huge market in the industry, and so on and so forth. Okay, what I want to do now before going to the main topic is to do a quick propaganda about what we can do in the lab, because it might be relevant to you that are working with materials in general. And this topic is entitled Single Entity Electrochemistry. In brief, all of you know that nanomaterials are tunable. They have large surface area. Typically, they are imperfect. That means that they're not atomically precise unless we're talking about nanoclusters. And because of that, it's hard to correlate sometimes between structure and property relations. It means that sometimes the bulk property is not the one that you see and observe in experiments. You actually look at the minority part of your population that does the catalysis or the reaction that you want to do. And for that, we're going and introducing single entity electrochemistry with the ability to probe one entity at a time. And when I talk about entity, it could be any type of object. It could be nanoparticle. It could be a molecule. It could be even a single atom. All of these were demonstrated in the literature. One of the motivation that my lab was focused or is still focusing is to take this culture counter technology back from the 40s that is still being used when it comes to counting cells and red blood cells, for example. And the idea here is that you just have a flow of your cells. Once they block the space between two electrodes, you see an attenuation of your current, and you can count them one at a time. And what we developed with other groups is the ability to do the same thing in a much straightforward experimental way with a resolution that goes down into doing 10 nanometer scale or even lower. And I want to share with you how it actually works. The way that it works is that you take an inert microelectrode. Typically, when you use electrochemistry, you use large electrodes for catalysis, for batteries. In this case, I'm talking about micron scale microelectrodes. And the physics and the diffusion in this case is a bit different than in the macro scale, and I will not talk about that. On top of that, you are adding into this solution with the electrode a dilute solution of, in this case, colloids or nanoparticles that can do some catalysis. When I talk about dilute solution, I'm talking about picomolar concentration. And what you have is natural diffusion of these catalysts in solution. And from time to time, they stochastically collide with the electrode surface, one at a time. That's why you need them to be diluted. Once they collide with the surface, they can catalyze some reaction that does not occur on the inert microelectrode. So I'm applying a specific potential that is insufficient to derive the reaction on the microelectrode, but once you have this electrocatalyst, it can derive this reaction. And then we measure the current. And I would like to share with you a real-time demonstration of how it looks like. If I can. There you go. On the y-axis, you're seeing the current. On the x-axis, you're seeing the time. We call this Chrono Amperometry. And what you're seeing onto your right is a cartoon of what we think is going on. You have a diluted solution of nanocatalysts, and every time one of them collides with the electrode, you're seeing a discrete step that corresponds to the electrocatalysis arising from each one of these electrocatalysts. What it tells you, or what it can give you, is information about the distribution of the activity of each catalyst one at a time. I want to share this slide and then move to the main topic, just to share with you a recent paper of ours that talks about how platinum nanoparticles, which are very, very relevant for hydrogen economy, for example, and some of the reasons and insights that we got from the single entity electrochemistry that tells you how do they degrade over time and what are the reasons that they degrade over time. And the main concept of why we're doing this is to try to correlate events that are happening at the nanoscale, at the single entity level, with macro scale devices.
So Lior, maybe you say that we can interrupt you. Maybe I can ask you a question here. Yeah.
Absolutely.
So first, a simple question. You said inert microelectrode. What material of the inert microelectrode you fabricate?
Fantastic. So the materials that you can use for the microelectrode are as abundant as the macro electrodes. The typical ones that are commercially available will be carbon fiber, gold, and platinum. Can you devise any kind of electrode? Yes. And you can do this in the clean room, for example, without any problem. The main limitation will be stability in solution and oxide growth.
Mm-hmm. Okay, so thank you. The second question related to how do you control every time just one nanoparticle get on the electrodes?
Great, that's a fantastic question. And this comes back to the fact that we are using diluted solution. So let's say that now I'm increasing my concentration of my catalyst here in solution. What I will see is that my frequency, the frequency of the events will increase and increase. There is a direct linear correlation with the concentration. At some point, if the concentration is too high, it will be a continuous event and I cannot see discrete events. That's why we need to use dilute solution. And if I'm seeing over time only one, then I can indeed say that this is only one catalyst at a time. To be more specific, now we have the technology to couple this to liquid cell TEM. I will not talk about this today, but it means that we can not only talk about it in words and indirectly say that it's one at a time, we can actually see one catalyst landing on the surface one at a time.
I see. Thank you.
Oh, and just to emphasize a bit more is that, well, it's not that we have only one catalyst in solution. We have a diluted solution with many catalysts, but because it's diluted, we can see this one at a time. I think that Alon has a question as well.
Yeah, what drives particle adsorption onto the surface? Are you attracting the particles, or is this characteristic of the collision kinetics of the particles in solution on any surface?
Fantastic question. So Alon is asking, why do they stick on the surface? And I think that you actually answered the question. A, it can be that they do not absorb. It means that they can bump and go away. In this case, the way that I will see that, give me just a sec. I want to try and sketch. I don't know if I can. I cannot. But in this case, what I will see is a spike instead of a step because it's a transient event. I can modify the surface in order to make sure that the chemistry promotes adsorption, and I can use migration. It means that I can use low electrolyte concentration in order to derive my charged particles. In this case, there is carboxylite, there is negatively charged surface of the nanoparticle that attracts the nanoparticle to the surface. And on top of that, in this case, it's actually natural absorption of the nanoparticle to the microelectrode with reasons that we do not fully know, and I can just wave and say van der Waals forces, but this is, of course, not a complete picture. I hope that this answered your question.
Yeah, you've probably got somebody on the faculty there that knows something about it, no?
Quite a lot, yes.
Yeah.
All right, from here I will talk about the main topic of today, which is protein electro assembly. I just want to emphasize that we're not talking about assembly of electronics, although we're doing this in the lab, but we're talking about using electricity in order to derive protein agglomeration and aggregation. And I think, and maybe this is going to be a bit over my head, but one of the main motivation is coming from this guy, Yaroslav Hairovsky, who is considered by everyone as the grandfather of electrochemistry. I'm saying grandfather because the father now is Ellen Bard in Texas, at least for many people. But the main point that and the reason that I'm sharing this slide is, well, he developed voltammetry in general, but on top of that, as a side toy, he used to work a bit with biological molecules, and he found out that you can look at the degradation state of proteins using electrochemistry, and only lately people started to follow up this kind of question. And I think that we actually elaborated quite a lot on his vision, and I hope to convince you that this is the case. Before going into the results and the setup, I would like to make sure that all of us are in the same line when it comes to electrochemistry. So for some of you, it's going to be trivial, but for some of you, I might clean up some misconception about electrochemistry. What you're seeing on the left graph is the potential that you apply on a working electrode. This is one of the 3 electrodes that you use in electrochemistry, and this is the electrode that you mostly care about. This is where the reaction typically occurs. You're applying this potential with respect to some reference electrode. That's your reference frame. And you're forming, or you're providing a waveform, which is triangle. That's why we call this cyclic, because it's a cyclic going from one specific initial value to another value and then back. And we do this over time. That's actually the wave function that we provide. And then the measurement itself is of the current that you're seeing as a function of voltage that you applied. And the interpretation that you're seeing here is a duck-shaped voltammeter. And I want to spend a little bit time to make sure that it is clear what you're seeing here. Typically, what people are thinking, or some people are thinking, well, this is where oxidation occurs, and this is where reduction occurs. This is incorrect, and that's what I want to clean. What we have is you start with a potential that is insufficient to do anything in solution. You just have almost zero current. When I'm saying almost zero, it's non-zero because you have some capacitance coming from the electrode-electrolyte interface. As soon as you're ramping up your potential, something happens. What happens is that you're starting to exchange charge between your species in solution, any type of species that can exchange charge, and the electrode. And the higher the potential that you apply, the more current that you're getting. This regime that I'm showing you here is the kinetically limited regime. This is the regime where the electron transfer or the charge transfer event is limited by the kinetics of this charge transfer. At some point, however, you're getting a maximum. The reason that you're getting a maximum is not because here oxidation occurs. Oxidation occurs all over this place. The reason it happens is because at some point, you're now becoming limited by another process. And this process is actually mass transfer. It means that at some point, you have depleted the layer near your electrode. In electrochemistry, we call this diffusion layer, in such a way that now not the kinetics is the one that's controlling the current, but actually the diffusion of species to the electrode surface. This is the reason that you're seeing this maximum, and then it decays away because this diffusion layer is growing and growing and growing over time, which means that the time that it takes for other species to diffuse to the electrode is taking longer and longer. The same principle occurs on the other way around. So it means that now you have this diffusion layer, now you can reduce your species and you have a minimum from the same reason. Now you're controlled at some point by the mass transfer of this process. All right, another thing that I want to note about electrochemistry, and the reason that I am so attracted to electrochemistry, is that the electrode can act as a surrogate for any donor or acceptor that you have in homogeneous chemistry and materials chemistry. What I mean by that is that you can use the electrode as an acceptor or donor, that's of course, and more than that, you can actually tune the energy levels. What I'm trying to say here is that you can use electrochemistry as a delta G meter because of the ability to change the energy levels of the electrode simply by applying a potential to the electrode with respect to a reference electrode. And that's the main thing that I want to explain in this slide. The next thing that I want to talk about and focus is on the graph that you're seeing here. Let me go step by step with what you're seeing. On the y-axis here, you're seeing now a different monster, and this is actually delta I. The reason that I'm writing here delta I and not I as current is because we are using here a different technique, which is not cyclic voltammetry, but rather we call this differential pulse voltammetry. In principle, it's very similar to cyclic voltammetry, but you can see here that you're seeing only the reduction part and not the oxidation, although you can do this in both directions. But the main point that with that technique, you can take away all the capacitance coming from the electrode-electrolyte interface by some calibration, by differentiating your voltammetry, and you can enhance your signal. And that will be relevant when you talk about biomolecules. The disadvantage of this approach is that when it comes to kinetic studies, it is not as good as cyclic voltammetry. But in our case, we mostly talk about the thermodynamics. In this case, this method combined with high resolution is way more robust and suitable for what we want to do. What you're seeing here is a differential pulse voltammogram of perchloride, so just a pH 3 solution, which means that you have high concentration, 1 millimolar concentration of hydronium ions and a platinum electrode. That's it. And you're seeing here two waves. The first wave is the hydronium reduction. This is a classical reduction process that you see over and over in the literature. Again, what you're seeing here is the diffusion limited event, and that's the first reduction wave connected with hydronium. The other part is connected more with water electrolysis that you're seeing here, and it goes on and on because this is a rather kinetically slow reaction, and plus you have 55 molars of water, so that's going to be a long one to have. But what I want to show here is that just to share with you how to read a differential pulse voltammogram, and the information that you can get from here is the formal reduction potential, or the thermodynamics, in this case, of hydronium ion reduction. And this kind of techniques is again used for thermodynamics investigation, kinetics, which I will talk less about, but it's very important because you can talk about even coupled reaction. You can talk about kinetics happening in solution with electrochemistry. Electrochemistry is not only focused on things that are happening on the interface. You can, in a very analytical way, understand the kinetics that are happening homogeneously in solution with electrochemistry. And the application is various. And most of the people are talking about device characterization, sensing, of course, surface, and very few works on biological science, which we will focus about. So what I talked about today is about, or what I tried to explain in the last slide was about proton reduction. In this case, the process that you're seeing is that you take a platinum, you take some species, in this case B, that is protonated, you apply some potential, you reduce the protonated species to form a platinum H species bound to the electrode and the free species deprotonated in solution. What you're seeing here is actually the half part of hydrogen evolution reaction, which is so important for energy-related devices. The second part is another hydrogen that is coming, another proton that is coming from solution, goes to the surface and liberates the other hydrogen to form hydrogen 2, H2 in solution, and that's how you make your fuel. The reason that I'm talking specifically about platinum, well, for energy-related application, it's not great because it's just expensive, but it's a wonderful catalyst. For us, it's a wonderful catalyst, and we don't care about the price. That's why we're using platinum as an electrocatalyst for proton reduction. Before going into the science, one last slide to make sure that we're on the same page when it comes to biology, because many of you are coming just from material science unrelated to biology, and just to talk about amino acids in general. And this is the general structure of amino acids. What I want you to remember here is that you have some carboxylic unit or carboxylate, depends on the pH, units on one side. This is the C terminus, H2N or H3N plus on the other side, and some residue. These residues are the ones that distinguish between one amino acid to another. And I want to focus here specifically on charged amino acids. What you're seeing here, aspartic acid, glutamic acid, histidine, lysine, and arginine. All of these are the only fives that are charged in biology. I'm talking about negatively and positively charged. And the reason that I'm focusing about that, because you're seeing here that they all have protons. So to reiterate what I discussed so far, we have two processes. One is a proton-coupled electron transfer on the electrode. And the other thing is that protonation in biology, in many cases, is fundamental to biochemical processes. That is connected to acid-base reaction, that is connected to proton channels, that is connected also to conformational changes. And the question that we were asking together, and I'm talking about the collaboration with Dan and with Mike Gordon, was, can we combine the two reactions to manipulate proteins? That's the hypothesis that we have in mind. And the first question that comes into mind is, well, is there any protein that we can use as a model system? What could be a nice model system that we can show that this hypothesis, or we can ask if this hypothesis is correct or not? And well, luckily, Dan is an expert in these kind of proteins, and a beautiful example will be reflectin. Reflectin is a protein that is located in the squid, in the skin of a squid, and is the main reason that you see the camouflage properties of the skin of a squid. In principle, what's happening is that, and I'm talking about now in nature, is that you have this reflecting protein that is rich with histidines. There is about 30 histidines positively charged in the protein. And by doing phosphorylation, you're actually neutralizing the histidines. And by that, you're overcoming the Coulombic repulsion, and you're moving from a monomeric proteins to an agglomerated assembled proteins. Why does nature care about that? Well, what happens when they assemble is that you have water exclusion. And when you have water exclusion, your refractive index changes, and voila, you got your camouflage effects. So this is the basic principle, biological principles behind the function of this protein. The next step, which is a essential step before we go into the electrochemistry, is to understand that this kind of biochemistry can be done in vitro, outside of the living organism. And the way that Dan did this and demonstrated this was by changing the pH of a solution containing these proteins and measuring the radius of the proteins. And what you're seeing here is that when you increase the pH, at some point you're getting into a pH that is neutralizing your imidazolium moiety. That is a simple, pKa chemistry here. Once you are at the pKa, pH 6.5, that means that 50% of the residues of the imidazolium are charged and 50% are not. If you go even higher, all of them are becoming neutralized. And that's what you're seeing here. Once you're going higher with pH, you're seeing the assembly process. You're seeing how the hydrodynamic radius is getting larger and larger, indicative of assembly. And the next question that we're asking, can we do this in a heterogeneous way? And I want to emphasize why I'm talking about heterogeneous. It means that we are trying to develop a new method that instead of doing this in a homogeneous way, which is typically instantaneous in solution, we can control what's happening because it's happening at the surface of the electrode instead of everywhere in the solution. And this is the results that we got. So first of all, before jumping over a big barrier, we started by doing a simple electrochemistry of histidine in solution with a platinum electrode, and we got the following voltammogram. This simple result was one of the biggest results that we got because it provided a proof of concept to the ability to even do this with a more complex biological systems. Let me explain what you're seeing here. And on the right, you can see the molecular structure of the each amino acid that you're seeing here. So let's focus on the first voltammogram. This is the y-axis, the current. This is delta current as a function of potential that I'm applying. In order to refresh your memory, take a look at here. You're seeing one wave, you're seeing another, and you're seeing a third. Unlike in the case of the hydronium alone, which is our control experiment, you're only seeing the hydronium and then the water electrolysis. So from that, we can conclude, because this is the only moiety in solution, that we're seeing here an additional wave that corresponds to the imidazolium directly reduced on the platinum electrode. More than that, the reduction potential, or more precisely, the formal reduction potential of this imidazolium is different than the hydronium ion. That means that we can selectively reduce this depending on the potential that we apply. On top of that, we have another small discovery is that, well, we don't need to use only charged amino acids. We can even probe the N-terminus, and this is the small bump that you're seeing here. The next natural step after working with single amino acids would be to go to a peptide before we're going into a full protein. So for that, we took a triple glycine, and we took, yes, I'm ready for a question.
Okay. So yeah, this is very interesting. So you can see both the hydronium reduction, and then you can see the proton associated to the imidazole.
Correct.
So on the other hand, the proton protonation deformation is a very fast equilibrium in the solution in the aqueous. So in that fast equilibrium, they come on or off, but you still can differentiate. That seems a little surprising to me.
That is correct, and this is for me was surprising. I think that I will have time to show you what is the thermodynamics behind this process and how you can differentiate that. Now, another thing that you mentioned here is that this is actually a reversible process. It means that after you deprotonate this, they will be protonated back in solution, and you can see this also from the voltammograms, which I'm not showing here. So I'm showing here only half of the picture. But the main point is that you can derive with electricity the direct reduction of imidazolium moieties in solution, freely diffusing in solution. Okay, going back to the peptide picture, which is kind of an intermediate before we're going into proteins, what we can see is that when we use a glycine, we have this single wave, and that corresponds to a similar wave that you see with the N-terminus alone. And in the case that you're using the same glycine, but the only difference is that you're switching from one glycine to another, to an histidine, now you're seeing this kind of a broad wave. So intuitively, you would say, okay, that's not interesting, it's just a single wave. But with careful deconvolution, and you can see this by the eye, hopefully, you can see that this is actually two waves coming together. This is something that is well known in electrochemistry. Once you have two redox waves that are connected together, they can overlap. And this is exactly what you're seeing here. So again, the red line that you're seeing here corresponds to the imidazolium, and the amide is the one that you're seeing here. The reason that there is a shift here, I will explain this later, but this is coming from the fact that the pKa value of the imidazolium free in solution or in a peptide is a bit different. If the pKa values are a bit different, that means that the thermodynamics and the reduction potential that I will see will shift a bit. The last big jump that we have is talking about the full protein. Before doing that, I would like to brag quickly about something that is quite simple but elegant that we developed. And this is what we developed. And I think this is very practical for biology application. When you talk about biology, in many cases, these proteins, peptides are super expensive. When you think about electrochemistry, you typically work with large volumes. I'm writing here 1 milliliter, but typically it's 5 milliliters or even, liters, depending if you're doing bulk electrolysis or just sampling something. What we were able to develop, and that took time, is electrochemistry on a drop. Now we have a much better apparatus than what you're seeing here, but the main point that we were able to decrease the volume of the solution that we have, still using true reference electrode, to about 10 microliters to 100 microliters, up to our will. And we took a lot of time to make sure that the thermodynamics that we're seeing with simple redox probe are the same as you're seeing in the bulk, but really comparing this one-to-one. And the reason that I was worried about that was because, well, simply from physical chemistry point of view, we're changing the boundary conditions here, and that could have an effect. So with careful measurements, you can really imitate what you have in the bulk, but do the same thing, but with low volume. Yes or no?
Is there a time limit on these experiments? Do you get drying of the droplet, or do you have ways to keep it hydrated?
That's a fantastic question. So what Alon is asking, well, great if the thermodynamics is correct, but well, the drop itself can evaporate with time. What I can tell you is that we specifically used 100 microliter here, so high drop, in order to minimize this effect. I have now a student that is doing a step-by-step experiments that will tell what is the cases where, what is the time frame that you need for each typical volume. So that's a kind of a rigorous, more boring work to know how the concentration here changed with time, when it comes to evaporation. But the bottom line is that if you're using a high volume compared to like a 10 microliters, this process is rather slow. The reason that I can say this with confidence is that, well, we can see that with the eye at least, that the size of the bubble here is staying constant. More than that, our experiments are typically happening or occurring over 5 minutes. But when you're using smaller drops, that effect is actually faster. I hope that I answered your question. And this is the big, sorry, I jumped one thing. In the case of reflecting, what we're seeing here is the classical hydronium reduction. And on top of that, we're seeing here a shoulder. Now, as a non-electrochemist, you can say, great, you're seeing this kind of small shoulder and you're gonna make from this an elephant. And I am not sure that I believe that this is actually real. What you're seeing here is, first of all, a deconvolution of these two waves. So that's one way to explain that this is indeed real. And the other one is to see that the potential that you're seeing here really corresponds or close to what you see for imidazolium reduction. But I would like to explain that the main reason that the peak here is small is because electrochemistry relies on diffusion. Now, so far we talked about small molecules. That means that they diffuse fast in solution. That means that the flux of these analytes to the electrodes is large. When you talk about proteins, now the signal that you're going to see will be extremely slow because the diffusion coefficient is so small. They move slowly in solution. So the fact that we were even able to see this kind of direct proton reduction with a full protein, for me, is mind-blowing. And by the way, this is the reason that we used differential pulse voltammetry, just to enhance our signal compared to cyclic voltammetry. If this is not enough, we do have a direct indication. In order to do that, what we've done is coupled electrochemistry to dynamic light scattering. What you're seeing here is a different apparatus. Now we're working with a macro scale electrode, and this is a coiled platinum electrode. And on top of this coiled platinum electrode, where we do the electrochemistry, we coupled dynamic light scattering, so we can really probe the species that are forming next to the electrode surface. So let's take a look at what we got. On the left side here on graph C, you're seeing the count rate, so the signal coming from the dynamic light scattering as a function of time. And we did 2 control experiments and 1 real experiment. The control experiments, and let's talk about figure C and D together. The control experiment was A, stability. So we used what we call OCP, open circuit potential. What it simply means is that you don't apply any potential between the in the electrode. It means that there is no current passing at the working electrode. There's no chemical reaction happening. And if you look at the size coming from the DLS, you're seeing that this is around 10 nanometer. This is a log scale. That's definitely very encouraging because the size of these reflecting proteins are around 10 nanometer. The second control experiment was to apply a potential that is below the reduction potential. Let me show this because this is important. Below the reduction potential of the imidazolium, but still reduces the protons. Why have we done that? When you reduce protons at the surface, and I'm not talking about the protein, now I'm talking about just hydroniums, the acid in solution that we have, that was in pH 3, that means that you are changing the local pH near the electrode surface. That means that actually we can derive this reaction indirectly by changing the pH. And if you remember, I showed you that Dan Morse was able to derive this assembly by pH alone. So it was important for us to understand, are we doing this indirectly by changing the pH locally near the surface or directly on the surface? And the answer is that we're doing this directly. How can I tell that? Because even if I apply the potential that is sufficient to reduce the protons in solution, nothing still happened here. This is part of the reason that we used acidic solution. However, once I'm applying a sufficiently negative potential here, minus 700 millivolts, after a couple of minutes, you're seeing a boost in the count rate, and you can then translate this into size distribution, and you're seeing how the monomers are assembled and going back into and stabilizes after some time, around 10 minutes, into sizes that are around 100 nanometer. Or in other words, I believe that we were able to demonstrate by coupling these two dynamic light scattering that indeed we can directly deprotonate full proteins in solution and make them assemble. One note here, why was that super surprising to me? Typically proteins are insulators. Now, if you have these moieties, these imidazolium side chains inside the protein, the electronic communication between them and the electrode surface is unlikely. It is connected to the tunneling barrier that you have between these moieties and the electrode. I don't think that we deprotonate all of the amino acids, all of the histidines. We do have an estimation which is around 5, and I don't want to talk about that, but the main point is that we were able, with few of them that are probably more accessible on the surface, to derive this reaction. And I believe that these results that you're seeing here are convincing enough to show that this is indeed happening. If this is not enough, it's always nice to see by the eyes. So what you're seeing here is the same experiment that I described before, but on the left and right, you're seeing how the solution looks like before and after applying this sufficiently biased potential at the platinum electrode. Before applying the solution is transparent. You have a diluted amount of your proteins, and you just see the platinum electrode coil. And by the way, this is where the beam of the DLS is transferring from. And this is how it looks like after 20 minutes. And what you're seeing here is how the solution is becoming opaque, indicative of this assembly of the proteins only near the electrode surface. Above it, away from the electrode, it's still transparent. So this reaction, of course, only happens at the electrode surface. I want to go something that might be interesting to you as material scientists, is that we can also do this with 2 electrode configuration, which is relevant for batteries and for applications and optical sensors or whatever you want. But the main point is that we've done the same thing with only 2 electrodes without any control coming from a reference electrode. And you're seeing here more or less the same thing. One of the reasons that it's actually going back to even a micron scale is because we applied a really large potential here. If that's not enough, what we did is a very trivial experiment. We just took a pipette and we took some sampling around the electrode surface, and there you go. This scale bar that you're seeing here, I apologize for that, it's 100 nanometers. So you're seeing this assembly in a TEM image that was taken by taking the sample near the electrode surface. From here, and I think that I will do this shortly, we had a lot of options to where to go, either by application or understanding more what's going on. And what we decided to do is to try to extend the scope of material, or in this case, amino acids that we can investigate. So this is more kind of a purely physical chemistry approach that I try to push for. So again, we validated our approach or hypothesis that we can combine both to manipulate proteins. This is the electro assembly properties that I talked about. But then the question is, can we generalize this effect to any charged amino acids? In this case, I'm going back to all of the charged amino acids that offered by biology. In brief, what you're seeing here is that you can do that indeed with all of the charged amino acid, in excluding arginine, and I will talk about this very soon. Each one of the amino acids that you're seeing here, the charged amino acid, produced a distinct, specific formal reduction potential that corresponds to the charged moieties that encompass this peptide. In brief, what I want to show you here is the following beautiful calibration curve that we got. On the y-axis, you're seeing the value of the reduction potential. This is the minimum that you're seeing in each one of the curves here, 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 and here. A side note, the blue line is the curve that you get after subtracting the clean solution only of protons that you see here as a dotted line, black line. So this is actually, the measurement is the gray line, and after subtracting this from the dotted black line, which is the control experiment, you're getting the blue result, which is your thermodynamic information that you can get from the system. What you're seeing here is that we found a universal correlation between the pKa, the basicity or acidity of your side chains, and your N and C terminal of your peptides, and the formal potential. So that's kind of a calibration curve or titration curve that you have between any type of acid that you want, biological acid, and your formal potential. So in other words, yes, we were able to show that this is a universal phenomenon that we see here, and we were able from now on to actually predict, show me a voltammogram, and I can tell you what is the pKa of your side chain in solution. And we're now thinking about doing this even in a more complex environment where it's harder to probe what is the pKa that is sometimes changing because of the internal environment. And this is very, very relevant for biological understanding of things that are happening, like acid-base reaction in biochemistry. I want to jump for a second about the arginine, which we were not able to resolve. And the reason that we were not able to resolve arginine is because the reduction potential and the pKa are so far away in this case that they coincide with the water splitting process. It means that now we have a strong background coming from the water hydrolysis. So we do have a limit here. And the limit is around 11.5 pKa, which is quite large. We have a range between 3 to 11.5 pKas, okay, that we can use for this method. One of the reasons that we can care about that is coming from lysine. And I think that with a few more slides, I will finish. What we're doing now is actually doing manipulations to another peptide. In this case, it's a homopeptide of just 10 lysines all together. And the reason that we chose this system is because lysine is known to convert between a random coil that you're seeing here, which you can think about as spaghetti structure of your peptide, to a alpha helix form that is happening when you have the deprotonated form. And this happens above the pKa of lysine, which is high, around 11, so that makes sense. And what you're seeing here is a circular dichroism measurement that shows you how the transition from a random coil here to alpha helix that you see here. So we were trying to ask ourselves, A, can we convert this structure of lysine or homolysine from a random coil to an alpha helix, even under acidic conditions? And more than that, can we learn about the thermodynamics and kinetics of this process in a way that can never be done in a homogeneous solution? And one of the reasons that I'm sharing this with you as material scientists is that, well, I'm very proud of the fact that we were able to couple our electrochemistry to a lot of spectroscopy. It's not only dynamic light scattering, it's not only UVVs. Now I'm showing you also circular dichroism as a way to understand what's going on. And in principle, and I just want to make it brief, this is the result that you're seeing. We're talking about pH 3, so acidic condition. This is where the lysine is in a spaghetti random coil structure. And this is the CD, the circular dichroism signal that you're seeing, which is the classical signal of a random coil. Once we apply use how this wave propagates from a random coil to an alpha helix, even though this is done under acidic conditions. The reason that it's happening in acidic conditions that we replace pH with voltage. This is kind of a full propagation of this time that I'm talking to you about. One thing that is interesting here is that the time scale for this process to occur is actually slow, around a few minutes. So this is also works favorably for us because we can monitor this quite precisely and average our measurement with time. What we are thinking about going with that is, I think that this method can allow us to understand how the kinetics of the conformation from an, in this case, random coil to alpha helix is actually happening. So we can deconvolute between diffusional processes, between deprotonation processes, and actually the transition between one conformation to another when we couple both electrochemistry and, in this case, an optical measurement, which is circular dichroism. I want to summarize my talk quickly and then share another slide that talks about the future of what my lab specifically without this collaboration is thinking about, with the hope that maybe this will be interesting to you as well. What I talked about today is that voltammetry allows us to analyze proton affinities, even in the case of complex peptides. In some cases, you can really probe is typically not conductive and does not communicate with the electrode. The main bottom line is that we can now coin a new term, which is Electro Assembly. If you remember the dogma that I showed you at the beginning, where you can use electrochemistry to power things, so you can use chemicals to provide energy, or you can use potential to make some chemical reaction, now you can see that you can manipulate systems such as biological systems with electricity. The last thing that I want to share with you is something that I'm very excited about lately, and it is correlated with biology as well. And this is connected a bit to the origin of life, and more specifically to liquid-liquid phase-separated system. And recently there is a big buzz about RNA-based and peptide-based droplets that are occurring randomly. These are dynamic droplets that are occurring randomly in the cell, and they have a lot of biological functionalities. Many of them are unknown, but it is clear that they are connected to neurodegenerative diseases. These are the kind of droplets that you're seeing here. In this case, it's just a demonstration from milk, but you can think about them as well happening inside the cell. And what I believe that with electrochemistry and with the single entity electrochemistry that I shared with you at the beginning, we can ask very simple questions here that could be very important for biologists. And these questions are, how many protons do you have in this membraneless? There is no selective membrane. So you don't know exactly what is happening inside and what exactly are the species that you have inside these droplets, and are they different than the bulk of the cell? So the question that we all want to ask is, how many protons do you have there? How many oxygen do you have there? And maybe how many water do you have there? The question about water was actually answered already by NMR. But oxygen and protons, which are all electrochemically active, can be probed here either in the bulk and one droplet at a time. I think that with that, I would like to acknowledge all of these people. First and foremost, Dan Morse and Mike Gordon. I want to say something personal here. When you start as an assistant professor, and this is the situation that I am, you start to deal with a lot of things that you don't want to deal with. I don't want to elaborate on that, but that's what you have, that's reality. I mean, this job is amazing, but there are some things that are not so fun. I think that for me, at least, this is the reason that I became a faculty. This wonderful collaboration that are just brainstorming with smart people that push you to do new science and new discoveries that keeps you awake at night thinking about that. With all of the help of Dan and Mike, that would never been happen without the beautiful hands-on work of Sheng Ping, who is now graduating from the chemistry department, Rob and Brandon, who did all the preparation of the reflecting, and Stephanie, my student here, that developed the single droplet electrochemistry. And now we have a new generation that will replace Sheng, and she's already working with us, Eloise. And of course, to the TEM facilities, wonderful TEM facilities, not as such as USI, in UCSD, which are Claire and Aiden. And I think that with that, I would like to thank you for your kind attention, and I would be happy to hear any additional questions.
Thanks very much, Lior, for a great talk. Looks like we already have a hand raised from Jibin.
Yeah, thank you, Lior. It's a very beautiful work. I think I really enjoy it. It's also very, it's very novel. I think this is the novelty is really impressive. So my question is still kind of related to the one question I asked you earlier, because proton transfer is a very fast process. Right? So proton transfer goes, I think usually people consider it like a diffusion limit. So I'm just curious. So you can reduce attach affiliated with whichever residue, the histidine or whatever. But after you reduce it, so once you get into out of the electrodes, get in the box solution, and your pH is still maintained. Did you monitor the pH with time?
What we're doing now is, yes, we're doing two things. And I did not share it here because it's rather kind of new, but I'm pretty confident in what we have. We did two things. One, which I did not share, is actually finite element simulations. That means that we can simulate the pH gradient near the electrode surface. On top of that, what we're doing now is using dye redox probes that can tell us what is the pH literally by direct measurements. What I can tell you is that, well, it depends on the potential that we can apply, but as long as it's not too high, we cannot go above pH 7. This is a superficial answer, but what I'm trying to say is that, yes, now we're focusing on really understanding the gradient of the pH near the electrode and what is exactly the contribution coming from that. I hope that answered the question.
Yeah, I think just to follow up a little bit. So this, the data seem to suggest after it reduced, and then when you go bulk, when we diffuse a bulk into box solution, they still retained as that form. So they won't be influenced by the environment. They only exchange it with the environment, because if they exchange it with the environment quickly,
okay,
then they just come in and out, and they still go. Average result in the bulk solution, the pH determines.
Now I see the question, so I did not answer the question. So two things. One of them, it's something that I did not speak about, is that you have a counter electrode. It means that you have a counter reaction on another side of the electrochemical cell. It means that in general, the solution itself is neutralized, but you have again, a proton a concentration gradient. What we can, let me try to share this quickly, because I think that will help. This is the solution. And once you shake it, this is after doing the voltammetry, once you shake it, it goes back into the monomers. This is reversible because the pH in the bulk solution is still acidic. So this is a reversible process. So only near the electrode and only once we apply a potential, we can maintain this assembly. If we stop the potential and wait, it will go back into a monomeric reaction. I think that this hopefully explains a bit more what's going on.
Thank you. Very beautiful work.
Thank you.
Joe?
Yeah, being from the UK, of course, I had to wait to the very end to ask my question. But yeah, no, the first thing is the light scattering ECAM setup is really nice. I think that's fantastic. And then, yeah, sampling the TM out of that is great. Yeah, my question is more about the structures. So in general, do you see any fundamental differences for structures made by electro assembly versus other methods? Or do you think it's more really about how you can initiate and where you can localize them?
I think that, I don't know how it is so fast, but this is exactly the heart of what we're trying to do. That will take, I think, a couple of years. Let me reiterate the question and then explain why is that so important, at least to us. The question is that, well, you have a homogeneous process by changing the pH, for example, and you have this heterogeneous process. Is the outcome of the assembly is different or not? The answer, and we know this from TEM, is that yes, there is a difference. It's not the same outcome. I showed you specifically these agglomerated beautiful pictures, but that's not a representative of what's going on. We actually see some star shape and more than that. The way that we tend to tackle this question is by introducing also controlled steering of the solution, by controlling the mass transport. Why? Actually, what controls this whole assembly is the contact. You deprotonate one proton, you deprotonate another. If they're accessible as deprotonation, they can come together and form assembly. So I can control the mass transfer, just simply steer the solution. And by that, I believe that we can control the assembly that you're seeing. More than that, besides the material part that is interesting that you can form by these different materials, you can think about early intermediates that are forming during important neurodegenerative diseases that can be maybe resolved from the first time because it's a heterogeneous process and not a homogeneous process. Thank you for the question. I'm very happy with that.
Thanks.
Alon, did you have another question?
Yeah, I guess one clarifying question and then maybe a follow-up. So when you're doing the deprotonation, is that happening at the electrode, or are you modifying the local pH of the buffer in the vicinity of the electrode such that the protein or the amino acid is deprotonated?
Got it. And for that, we have a clear answer. And the answer is that we directly deprotonate some residues directly to the electrode surface. How do we know that? Because we can apply a potential that is now controlled by mass transfer of the hydronium ions. It means that we are changing the pH and we're seeing that we do not get the assembly. And only when we apply a potential that is sufficient and corresponds to the direct deprotonation of the histidines, we see the assembly process. And on top of that, we have simulations as well.
So I do have a follow-up then. Do you think this is generalizable to arbitrary proteins? So the reflectant is highly charged, yeah? So it's not going to tend to stick on the surface, even if you deprotonate some of the histidines. But other proteins are very prone to denaturation and adsorption on the surfaces, especially if you start getting rid of some of those ionizable charges. So do you have any speculation as to what types of systems or how you might avoid that while still maintaining charge transfer for this applied voltage mediated assembly?
That's a great question. And one of the things that are overlooked in electrochemistry in general is the fact, and people are avoiding that many proteins are indeed denatured at the surface of the electrode. So my answer will be, unfortunately, that no, I don't think that could work for a lot of proteins. What I'm hoping that it will work for proteins that are relevant, for this example, for neurodegenerative diseases such as tau proteins. I don't have at the moment the ability to predict if that will work or not. And you are correct that this is a main concern, because if they degrade at the electrode surface, it doesn't matter if I deprotonate it or not. I'm not looking at the process that I want to look at. The nice capability that we have here is that we can start to talk about coupling this with high resolution spectroscopy and microscopy, and by that to try to understand if that's really happening or not. I hope that at least partially I answered your question here.
Yes, yeah, thank you. And I'll echo Zibin's comments. This is an amazing experimental setup, and you can get some really unique insights here. So this is really great.
Thank you. I appreciate that. And yes, if there's any postdocs in the crowd and students, please, I think that I heard only PIs, but your mind is less contaminated than ours. So please feel free to ask any questions.
Hey, Lior, I have a question. It was a very great presentation, by the way. Coming from a biotechnology background, I am fascinated by the electron transport chain, and they do perform kind of similar roles. Like the electron transporting from one enzyme to the other. So I was thinking if you could observe the effect of this potential application of potential difference on the conformational chain in those enzymes, if that's
okay. So let me decouple between what you're asking here. When you're talking about electron transfer chains and you're talking about, for example, photosystem 1 and cytochromes, these are typically systems that are not related fully to proton reduction, just electron transfer. These systems have been demonstrated in the literature that you can probe the electron transfer events, although some of these works are problematic. But I think that what I'm doing here is a bit different. I'm talking about deprotonating this system. So they're typically not relevant, I hope that I'm not mistaken, with electron transfer per se, in order to change the structure of the protein. I hope that was a good enough answer for what you asked.
Yes, that's what I was wanted to know. Thank you.
Questions from other students? Yes, go ahead.
Hi, Leo. I'm a postdoc here. It's a very nice talk. So actually, I have several minutes internet cutoff. I'm not sure if my question has been asked, but I think I remember in your page 25, you show a very nice linearity between the pKa value and also the potential. I'm wondering if all these scattered point was done in a uniform bulk pH, or is it in a different pH?
Fantastic question. Let me try to find where it is, the calibration curve that we have. I don't see it. Why I don't see that? Just a sec. And the answer is yes. All of them were done under pH 3. So all of them were done under the same pH.
Okay, I see. In this way, do you think if the bulk pH will have certain impact on the linearity of this?
Absolutely.
Of the system?
The answer is straightforward, yes, but it's predictable. This is coming from what we call in electrochemistry Nernst equation, and you should have something like 59 millivolt offset. It's coming from 1 proton, 1 electron reduction. I don't want to talk about it. If you want, we can talk about it later. By the way, the slope that we got, this experimental slope, is around 50 millivolts, which is very close to this theoretical value. For me, it's good enough if you're talking about this kind of repertoire of different amino acids. But yes, if you change the pH, this whole thing will shift.
Okay, so my second question is more general about the electro assembly, as you mentioned at the end of your talk. So by applying a potential, you're kind of changing the protonation state and also the charge of the amino acid section, which can also be done by tuning the pH. But by doing electro assembly, so you are using electro potential at a single pH to realize this. But for the electrochemical assembly, is there any advantage over the normal, like the pH tuning, and to change the transition state of the assembly?
Got it. And so that's a great question. And I do have, I think, a clear answer about that. And this is what I want to emphasize. The advantage is heterogeneous process versus homogeneous process. It means that instead of doing this everywhere in the bulk, you can do this only near the electrode surface, and you can control this process in a very fine-tuned way by simply applying different potentials. And the impact, the possible impact of that is that you control the structure of these formed assemblies by potential in a very controlled way, only near the electrode surface.
Okay, thank you.
Other questions? I found the in situ CD measurements during the experiments really impressive, by the way. I don't know if there were specific challenges with keeping everything stable enough. I'm sure there were.
Yes, there were, but all the credit comes when it comes to this design is to Shen Ping and to Mike Gordon from chemical engineering.
Okay, really, really impressive. All right, if there are no further questions, let's thank Professor Sepunaru again with our virtual applause.
Thank you, everyone. That was extremely enjoyable. I hope to see you in person soon.
I'll see you shortly. Thank you.